Tala Falava, and welcome to our next Year 12 biology lesson. But before we start today, let's recap on what we learned the last time we had our lesson. Do you remember? You're correct. Last time we talked about the flower and the different parts of a flower. We also talked about the process of pollination. Pollination is a process which the pollens are transferred from the anther to the stigma. And lastly, we talked about the different types of agents. These agents, they help speed up the process of pollination. Do you remember an example of an agent we talked about? You are also correct. Yes, we talked about bees or insects and birds. So that was our previous lesson. Today's lesson, we will also focus on strand 4, which is plants. Today, we will be focusing on identifying the internal parts of a leaf. So when we talk about internal, we are talking about inside of a leaf. Secondly, we will describe the functions of these internal parts of a leaf. And lastly, we will look at describing the function of a stomata. But before we go into the internal parts of a leaf, let's talk about leaves in general. I have brought these two plants in to look at the different types of venations of leaf. So when we talk about venation, do you understand what the word means? Venation, we are talking about the different types of veins or the type of vein structure that the leaves have. So I bought this lemon tree, and if you look closely at a lemon tree, their veins are quite network-like. So they look like a net. So this is why this type of venation is called network veins. The second type of venation, we are looking at parallel veins. So if you look at this plant, they are quite long leaves, and their veins are in a straight line. That's why these leaves are called parallel vein leaves. So if you look at the difference between the two leaves, this is a network veins, and this is a parallel vein. And it's easy to identify the difference between the two by also looking at their root structure. So network leaves, they normally have taproots. So taproots, they have a long main root. So you see here, and then they've got a lot of root hairs on the side to absorb all the minerals and water. And there's a main root. This is called a taproot. With the leaves, like I've mentioned before, their leaves are network leaves. So network leaves with taproots. But if you look at our other type of leaves, these are parallel leaves, and they have fibrous roots. So fibrous roots, you can see the difference. They don't have a main root, but they have many root hairs on the side that come from the main bark of the tree. So you can see the difference between the two. So in general, if you look in your garden, you can pretty much predict the type of roots the plants have by looking at their leaves. If a plant has network leaves, they pretty much have a taproot. They have taproots. But if plants have parallel leaves, they normally have fibrous roots. So that's part of your investigation and observation that you can do at home. So remember, parallel leaves, network leaves. the two different types of leaves. They are called venations, so different types of veins, structures and leaves, parallel and network. And then you look at their roots, tap roots and fibrous roots. Parallel leaves, or the plants with parallel leaves, they normally have fibrous roots. Network leaves, the plants that have these type of leaves normally have tap roots. Awesome. So this is the external parts of the leaf. But what we're talking about today is the internal parts of the leaf. So inside of the leaf. Okay, I'm sure that this is revision for you. So I'll give you a couple of seconds to draw a picture of your internal part of the leaf. How are you going with your picture? That's awesome. Let's look at the picture I'm going to draw on the board. I've outlined the upper and the lower epidermis for us. So always remember, when you're drawing an internal part of the leaf, always make sure that it is clear so you know the difference between the upper epidermis and the lower epidermis. So on the top, so this is the upper epidermis. Upper epidermis, and this is the lower epidermis. On top of the upper epidermis, we always remember that there is a waxy cuticle. So the waxy cuticle, it sits on top of the upper epidermis. Cuticle. Below the upper epidermis, there should be, yes, chloroplast. So there are a lot of chloroplasts here in the upper epidermis. Below these compacted chloroplasts, there is a lot of space. So there is a lot of space below here. So this is another chloroplast just floating around. And also down here at this layer, there are your tissues. Do you remember the two types of tissues that plants have? Yes, there is your xylem and your phloem. So this is your vascular bundle. So these are your vascular bundles. So waxy cuticle, then you have your upper epidermis. The upper epidermis, there are your compacted chloroplasts. Remember, these are layers. These are two different layers. Your top layer is called the palisade mesosphere layer. Palisade mesosphere layer. So e longo fia lava le palisade mesosphere layer, e pizzo ai lunga o le internal parts of the leaf. Ai se ai pizzo ai lunga, o wao I tonu lava o lo tele ai le chloroplast. Chloroplast e tele ai le pizzo ai lunga laula au. Why do you think it's a wana tele le chloroplast I le pizzo ai lunga laula au? Yes, because it is easily exposed to the sunlight. Remember, sunlight is do you remember the word? It's a catalyst. So sunlight is a catalyst for photosynthesis. E vave ona ngai yoi ma tupu le process le photosynthesis pe afa pe e yai le la. O le mea tonu lava na, and that's the main reason e te le chloroplast le pizzo I lunga o le laula au. The bottom part is called the spongy mesosphere layer. So ila le spongy mesosphere layer e kau le yaingi chloroplast e vava mamao, and there are lots of air spaces. E whawhaingo fie on the exchange ya gases. Wa gases ya le exchange or the carbon dioxide, ma le oxygen. So that's why it's not as compacted as the palisade mesophilea, o lalo I le spongy, e whai o le vava mamau o chloroplast, ai koe maua iai, ma le tato tissues e lua. There are two main tissues in plants. We have our xylem, and our phloem. Do you remember the function of the xylem? Yes. Xylem is a tissue transfers water or transports water throughout the plant. Ole phloem o ia transfer ai lea. Yes, mea ai o mea ai o minerals around the plants. So both of the xylem and phloem e maua tonu le vescala bando. Le maua lale lea maua le vescala bando e maua I le spongy mesophilea. The bottom where the lower epidermis is, there is the opening of the stomata. So the opening, po le ava noa le tonu I, this is the stomata. I control the stomata I le taponi maltatsala o lea guard cells. So these are your guard cells. So the guard cells, they are the reason why the stomata is open. Why? Why do you think the guard cells open? Ua matsala yele the guard cells ona ua turgid. Always remember that word. Who's heard of this word before? Turgid. O le winga o le upu turgid, ua tele le vai. Ua tele le vai totonu o le la au. O le laula au wa mafu ai ona matsala le stomata. E matsala stomata ina ia mawhai ona release le vai because with too much water. Just like us, when we have